Hello class. I continue on chapter 16. I think that this is going to be, the third segment of the chapter 16. So we have talked about DNA structure and what the Watson, Crick, and then Maurice Wilkins and the Rosalind Franklin did to they organize their data and then finally define the double helical structure of DNA and also how the base pairing works and then how the those bases gonna be going inside of the helical structures. So we are going to talk about in here is that I don't think we are going to talk about the details of DNA replications, but what we can talk about in here is that how they come up with this copying idea, the possible copying mechanism, how DNA is synthesized in the cell. That was actually predicted by Watson Crick's first paper in 1953. Actually, the copying mechanism in chemistry, in making polymer, is not really new, but how that is working in the genetic material was the one of the questions. So, the basic principle is that, okay, when they make the new DNA, and then probably they will copy the existing DNA molecule. And then we know that DNA is double helix and anti-parallel. So these two DNA strands are complementary. So which means that they have to somehow, unwind, okay, separate it, and then each strand will be work as a template, isn't it? And then they'll build upon a new strand using this two existing strand, and we call that as a replication process. And then they have to follow, base pairing rules, A, T, and G, C kind of rules. So that actually automatically sends off some kind of so-called semi-conservative models, but we still, were not sure at the time that really semi-conservative model was, correct or not. So let's say this is the parental molecule, and then when DNA replication of copies to occur, and then the separation of the strand occurs. So it's a complementary, complementary and then anti-parallel. So here, this is 5, and then 3 prime, then this is the 3 prime, and then 5 prime. So 5 prime, 3 prime, and then 3 prime, and then 5 prime. They are separated. So new strand will form here, isn't it? So isn't it? Also here. So something like this. Okay. So that is what you see over here. So T, G, A, T, C. So in here, last time it was 5 prime and then 3 prime, right? So probably here is the 5 prime and then 3 prime. So here is the 5 prime and then 3 prime. So new one is here's the 5 prime and then 3 prime. So A, C, T, G, A, G. So it will form this kind of newly synthesized DNA. So that is the we'll say semi-conservative, because newly synthesized DNA is a hybrid of already existing DNA. So Watson and Crick predicted semi-conservative model, but somehow next, people has to, demonstrate that semi-conservative model is correct or not. So, next group of, people kind of come up with the idea that, okay, whether the semi-conservative model is or not, so they have to come up with a different possibilities. So that is conservative model and then dispersed model. So conservative model is that, okay, the newly synthesized DNA is do novo, somehow, newly synthesized DNA will never mix with old, template DNA. So in this case, old DNA and the newly synthesized is always separated. And semi-conservative is that, okay, you will always have the hybrid, but you will have the also newly synthesized DNA as they repeat their DNA synthesis process. And another one is the dispersive model. So somehow you can think of the DNA will be chopped up, and then they will be copied and then reassembled. Not the linear fashion, but they can be divided into many different pieces, and then they can recombine together later. So this is the another possibility that they can think of. So this is what we call as a dispersive model. Okay, so it's like an evenly distributed. Okay, every time they replicate the DNA. So Matthew Messelson and then Franklin Stahl, they supported this conservative model, and then they come up with the idea that, okay, all the strand was labeled with a heavy nitrogen, so that is N15, okay, and then new nucleotide was labeled with a lighter isotope, that is N14. So you grow the bacteria in like a first generation or something, okay, and then you isolate their DNA, and then the heavy DNA will be the original DNA, parental DNA, and then the newly synthesized DNA is the light DNA. The light DNAs. So whether they are mixed or not, or evenly mixed, or only hybrid and then intermediate. So that's what they are looking for during this kind of experiment. Okay, so here, so how they did is, okay, so they already bacteria already grown with a 15 N, which is the heavy isotope. Okay, and then later on, you put those cells into here, and then this is a new media that contains the lighter isotope. So what will happen is, okay, when the DNA is replicated, before the replication, they only show single band. But what will happen is, okay, they replicate, and then they replicate in this lighter condition. And then if they isolate DNA and then run it through the centrifugation, then you have less dense and then more dense, which is the OSH lighter, and then heavy DNA will be show up. So this is lighter. So they on the top layer. So this is the new DNA, and then probably this is old or hybrid. Okay. Okay. So if conservative model is correct, then you will see two band, lighter band, and then heavy band, because there's no mixture. But if if a semi-conservative is the correct, then in the beginning, they will have a heavy DNA, but after one round of replication or two rounds of replication, you will have the two band shows up. So a little bit heavier band, which is the hybrid. Hybrid means that one strand is lighter DNA, and then the other strand is heavier DNA. And then you have lighter DNAs, which is the new DNAs. And then this hybrid DNA is lighter than heavy DNA. Okay, and then how about dispersive model? So dispersive model is, okay, they will be evenly distributed, chopped up. So what will happen is that they are going to be evenly distributed. So you will have only one band, which is the hybrid, evenly mixed, mixed, those band. So the result was, okay, what they found was this semi-conservative model. So semi-conservative model was the one that being supported by this Messelson and then Stahl's experiment. Okay, so we'll just take a look at the DNA replication. So DNA is a copying mechanism, and then they are doing it really, really fast. And then they have a very, very small error rate, okay? And then I'll continue that later. Thank you.